photosynthesis. This one is very big chapter and important, is it? So from the previous class that yesterday was held, in that from that portion, do you have any question, any doubt? Have you studied later on at home? Hello, anyone? Yesterday. So from there, do you have any doubt? This was that also known as C3 cycle and C3 plant, is it? So today we will learn another cycle that is known as C4 cycle. C4 cycle. This is also C4 cycle and C4 plants. Okay. The C4 cycle is also known as hatch and slack cycle. Hatch and slack cycle. Okay. This one is that you remember that in the Kelvin cycle, that the in Kelvin cycle, which operate within the uh, within the stroma of the chloroplast in mesophyll cell, is it? It operates in a stroma of chloroplast in mesophyll cells. This is chloroplast. Okay. Now, what happened in the C3 cycle, that the main enzyme is what? In the Calvin cycle, main enzyme is the RuBP carboxyoxygenase. RuBP carboxyoxygenase, that is Rubisco, is it? This is the enzyme for Calvin cycle. This one help to absorb carbon dioxide. And I told that this is the name as carboxylase and oxygenase, means the same enzyme can absorb oxygen also. Okay? Can absorb oxygen also. So when it will absorb oxygen, then the photosynthetic productivity will reduce, because for the sugar production it is required the carbon dioxide, not the oxygen. But this enzyme can also combine with oxygen. So in and when it absorbs oxygen, there is another process which takes place, that one we call photorespiration. And when photorespiration occurs, then photosynthetic productivity is reduced. Okay, so this photorespiration we learn later on. First, we will learn today that in today is C4 plant or hatch and slack cycle. Some of the plants are there like sugarcane, maize, sorghum, etc. Some other plants are also there. In these, in addition to C3 cycle, there is another cycle operates. Means C3 plus another one cycle. All right. Okay. Before that, we have to learn one thing about the special type of anatomy which is found in this plant leaf. These are all, see, these plants are monocot. Of course, the C4 plant takes the C4 cycle takes place in some of the monocot and some other dicot also, but mostly monocot. In this plant, what happens that in their leaf, say this is the leaf vascular bundle. In the leaf vascular bundle, surrounding the leaf vascular bundle, there are big cells which are found in the form of a ring, maybe one ring or two ring or more. That is, cells are around the vascular bundles are present in the form of ring. This arrangement, a special arrangement in the leaf around the vascular bundle, this type of anatomy, internal structure of the leaf is known as cranch anatomy. Okay, this is known as cranch. Cranch means, literal meaning of the cranch is wreath. That is the uh, circle or ring of cells or flowers. Okay, so wreath, this is circular. So here, this around the vascular bundle, there is one or more ring of cells. These cells which is surrounding the vascular bundle, this one is vascular bundle, and surrounding these cells, they are known as bundle sheath cell. Okay, they are called bundle sheath cell. These bundle sheath cells are large. They are large cell in compared to other mesophyll cells. They are large in compared to other mesophyll cells. And in these cells, large number of chloroplasts are there. Okay, many chloroplasts are there. But these chloroplasts are slightly different. In this chloroplast, that grana is not developed. Instead, there are the this thylakoid membranes. They are not organized into grana. That is, grana is less. A stroma portion is main. A stroma is there. These things are known as peripheral reticulum. So anyway, you should only know that these chloroplasts have very less grana. Grana is less. And there are mesophyll cells. In the mesophyll cells, the vascular chloroplasts are normal. Okay. Here the chloroplast also is large. This anatomy is cranch anatomy. What are these plants where we can see this cranch anatomy? These are commonly sugarcane, maize, sorghum, etc., panicum, panisetums. There are many. In these cells, so in this leaf, we have two types of cells. One is that mesophyll cells and bundle seed cell. Okay. Mesophyll cells, they have well developed chloroplast. Well developed chloroplast, and in those chloroplast, their chloroplast contain the grana. Here in the bundle sheath cell chloroplast, grana is poorly developed. Grana poorly developed. Now what is happening? Actually in this cell, in this plant, the carbon dioxide is absorbed by carbon dioxide is accepted by a compound which is known as PEP. PEP stands for phosphoenol pyruvic acid. This is what? Phosphoenol pyruvic acid or pyruvate. This is a 3-carbon compound. This one is 3-carbon compound. This 3-carbon compound PP absorb carbon dioxide plus water, of course, absorb carbon dioxide and produce a compound which is a four carbon compound that is known as oxaloacetic acid. This is, what is this? Oxaloacetic acid. Okay. This one is a four carbon compound. So what is happening? The compound that one absorb carbon dioxide, that is different. It is a three carbon compound PEP and form the oxaloacetic acid, which is a four carbon compound. So first stable product is a four carbon compound organic acid. And this is why that this cycle is known as C4 cycle. In the Calvin cycle, we have learned that the first stable product was a three carbon compound. So it is C3 cycle. Here the first product is a four carbon compound. So it is C4 cycle. This oxaloacetic acid is then converted into malic acid. These are also four carbon compound. This malic acid may change into that into another organic or C4 acid. Other C4 acid also it forms, but this one is the main. This malic acid, then what happened? This move from here to the bundle sheath cell. Again, it is here. Move to a bundle sheath cell. Malic acid here break to some three carbon compound, and when it break, then it release carbon dioxide here, and this forms a three carbon compound. This is actually the pyruvic acid. This pyruvic acid then transported to this mesophyll cell. It changed into pyruvic acid. That is transported. Pyruvic acid is then converted into phosphoenol pyruvic acid here. Again, when the phosphoenol pyruvic acid is formed, this phosphoenol pyruvic acid can absorb another molecule of carbon dioxide. Now what is happening? This carbon dioxide which is released in the mesophyll cell, in the bundle sheath cell, here this carbon dioxide is absorbed by RUBP and form two molecule of phosphoglyceric acid. Phosphoglyceric acid is converted into phosphoglyceraldehyde, phosphoglyceraldehyde to sugar, starch, and from some molecules is converted back to that through the regeneration process again RUBP is produced. This one is C3 cycle. So sugar formation or carbohydrate synthesis is takes place by the C3 cycle only, C3 cycle or Calvin cycle. Okay, the cycle which operate, the cycle which is operating the the mesophyll cells and the bundle sheath cell, this cyclic pathway is known as C4 cycle or Hatch and Slack cycle. So here in this process, in this plant, this C4 plants, there are two cycles are operating side by side. One is that normal Calvin cycle or C3 cycle, and another one is C4 cycle. C4 cycle operate between mesophyll cell and the bundle sheath cell. But the C3 cycle operates only in the bundle sheath cell chloroplast. In this, here what is the advantage of this cycle? This cycle has one advantage is that this PP, okay, this one is purely enzyme here, that PP combined with the carbon dioxide enzyme is PEPcase, means PEP carboxylase. Enzyme is PEP carboxylase. This PEP carboxylase is purely carboxylase only. So it will help to absorb carbon dioxide, not oxygen. So whatever amount of oxygen is here, this one will not support the oxygen absorption. It will help in carbon dioxide absorption. So this will carbon dioxide is passed to the Calvin cycle. And here what is happening in this bundle sheath cell, as this process is bringing regularly carbon dioxide here, so concentration of the carbon dioxide increases, and due to this high increase in carbon dioxide concentration, that photorespiration does not occur. Means here that Rubisco will not absorb oxygen because enough carbon dioxide is there, is it? So this photosynthesis or conversion of reduction of carbon dioxide will go on happily. So enough sugar is produced. In the mesophyll cells, chloroplast, there is no Rubisco enzyme. No Rubisco enzyme in mesophyll cell. Okay. So as there is no Rubisco here, so this will not absorb oxygen also. Neither carbon dioxide nor oxygen. For the carbon dioxide, PEP, carboxylase, etc. are there. This one, so this is the process. Due to this, that the productivity in this plant is very high. Photosynthetic productivity is very is high here. Where? In C4 plant. Hello, just a minute, please. I have an important call. हेलो. हाँ, बोलिए बोलिए सर. So sorry for the interruption little bit. Uh, okay, student. So in the C4 plant, the photosynthetic productivity is very high because there is no photorespiration. And these plants are actually, these plants are, they are tropical plant, mainly plants that grow in high temperature and high light intensity. And it is not affected by, and not affected by high oxygen concentration. Okay. So here I will show you. I have that in your book, one nice diagrammatic representation has been given, and I will show you that one. Okay, this is I will show you this diagrammatic representation of the photorespiration entire process from this. Okay, here you can see this diagrammatically in this. two two these two these cells have been shown. This is the cell of, here you see, cell of the mesophyll cell. This one is mesophyll cell, and this is bundle sheath cell. See here written that this is bundle sheath cell. If you cannot say, this one is bundle sheath cell, and this one is normal mesophyll cell. Mesophyll cell, you understand, that is the ground tissue of the leaf. In the mesophyll cell chloroplast, what is happening, as I told, there is atmospheric carbon dioxide is there. This carbon dioxide, this attached with the mixed with water, say bicarbonate is there. This one is absorbed by phosphoenol pyruvate. This is showing phosphoenol pyruvate. This phosphoenol pyruvate combines and they form a C4 acid. What is that C4 acid? I told malic acid. Is it? Oh, first one is oxaloacetic acid, malic acid, or it is aspartic acid also is produced. So all are C4 acid. These are C4 acid. Okay. So here this phosphoenol pyruvate combined with carbon dioxide in presence of PP carboxylase. That's written here, the fixation. So that forms the C4 acid. The C4 acid is transported through the plasmodesmata. Plasmodesmata are the cytoplasmic connection between the or among the neighboring cells. Okay. So this C4 acid, like malic acid, aspartic acid, that is transported to bundle sheath cell, and from the bundle sheath cell it enter in the chloroplast of the bundle sheath cell. Here the C4 acid break. Okay, C4 acid break into carbon dioxide and C3 acid, C3 compound, because one molecule, one atom of carbon is going out. So only three carbon atoms remain. So three carbon compound remain and carbon dioxide is released. This released carbon dioxide is absorbed and fixed in the Calvin cycle. Say this one is written here, that Calvin cycle. Okay, fixation by Calvin cycle. And then this C3 acid is then again transported back to the mesophyll cell, and here it regenerate the phosphoenol pyruvic acid. All right, and the phosphoenol pyruvic acid again absorb carbon dioxide. So what is happening? This phosphoenol pyruvic acid, this one absorb the carbon dioxide in presence of the enzyme. Remember the PEP case. That is the PEP carboxylase, and PEP carboxylase is not affected by oxygen. So it will never absorb Phosphoenol pyruvate will never absorb oxygen, only carbon dioxide. So the rate of photosynthesis is very high, productivity is very high. So this way, within the two cell, this cyclic pathway that operates, okay, that one is the C4 cycle or hatch and slack cycle. Actually, say this hatch and slack cycle is not for the synthesis of carbohydrate. This one C4 cycle is not for carbon dioxide reduction. That is not for synthesis of carbohydrate or sugar. But what it is doing actually, it is just working as a supplier agent. Actual carbohydrate factory is Calvin cycle. Calvin cycle or C3 cycle is the factory where the carbohydrate is synthesized. Okay, carbohydrate is synthesized, and the C4 cycle, this one hatch and slack cycle, is functioning as a supplier. They are the broker or dalal, or you can say they are the supplier agent who supply the raw material. Factory is there in the bundle sheath cell, which is the Calvin cycle, and that this one is absorbing here and releasing at the site of Calvin cycle. Okay, and again coming again, that in the factories also, the factory owner, okay, factory owners or their workers, they don't go for collecting the raw material. Instead, they have some agent supplier, they bring the raw material to the factory. So here the C4 cycle is only a agent or supplier of, and as the supply of the raw material is good, so the production of the factory also is good. So due to the C4 cycle, the C4 plants are they are having high efficiency in high efficiency in that carbon dioxide reduction. Okay. So in these two things operate in two different types of cells. Actually, what is being done here, that the Calvin cycle, C3 cycle, is affected by oxygen concentration. All right. So, in this, by this Cranch anatomy, by Cranch anatomy, the plant is keeping the Calvin cycle, keeping this Calvin cycle away from the site of oxygen. Where oxygen is there, this plant have separated it. There has been a barrier between the oxygen and these cells, Calvin cycle. This bundle sheath cells also, they have very thick cell wall, and in this, through this cell wall, usually the gas etc. is not diffused. Instead, the carbon dioxide is brought here through the cytoplasmic connection, that plasmodesmata. So this plant, they can grow in high temperature, that is in the tropical areas, that is at high temperature, and bright sunlight. Okay, and not affected by concentration of oxygen. So this is there are some advantages. This is an actually an adaptation against high temperature and bright light. Okay, and the oxygen concentration. So these are the here usually the question can be asked that first thing, that the what is this C4 cycle, and what is C4 cycle, and what are the plants and the C4 plants. Okay, the in C4 plants, another important thing is what is cranch anatomy. Then a small question can be asked like where C3 cycle operate in C4 plant. Ah, then characteristics or importance of C4 plants. Okay, these things can be asked, and overall reaction of the C4 cycle. I think here it is in a very simplified way. The actual is that I have shown here is the one. Okay, one thing here that I told that after after the releasing the carbon dioxide in the bundle sheath cell, this C3 compound is pyruvic acid. Okay, This pyruvic acid come back to the mesophyll cell, and this pyruvic acid change into converted into phosphoenol pyruvic acid. And here, during this conversion of pyruvic acid to phosphoenol pyruvic acid, ATP is ATP is consumed. ATP is consumed and AMP is produced. So, about for every molecule of pyruvic acid to convert into PEP, almost 2 ATP equivalent energy, 2 ATP equivalent energy is spent. So, here in the C4 plant, the expenditure of glucose or the carbohydrate production is high. Means for every carbon dioxide molecule to transport to the site of the carbon dioxide reduction, it requires additional 2 ATP energy. So total expenditure, if I say the total expenditure is now increase, and the total expenditure is coming to, in the C3 plant, for every carbon dioxide molecule there was 3 ATP and 2 NADP NADPH. Is it? That was the requirement. But here in this plus 2 ATP means it require 5 ATP and 2 NADPH. So expenditure here is more. And this for one molecule of glucose or fructose to produce 6 carbon dioxide are to be reduced. So if you multiply this with 6, then how many? That is 30 ATP and 12 NADPH is required. So production cost has increased here. But what is the problem? Because they are rich and they can spend that additional thing. They can bear the additional cost because this plant usually grow in a bright sunlight and high temperature. So they are exposed to enough light energy and they produce it and spend on their production of carbohydrate. So here we have to learn the difference between the C3 and C4 plant. C3 plant and C4 plant, what are the difference? If we go the first stable product, what is first stable product is PGA, phosphoglyceric acid. This is 3 carbon compound. And here the first stable product is oxaloacetic acid, a 4 carbon compound. What is the primary acceptor of the carbon dioxide? This is RUBP, that ribulose bisphosphate is the primary acceptor of carbon dioxide. Here, who accept the carbon dioxide? Here, PP is the acceptor. PP is the primary acceptor of carbon dioxide. Okay. Then the enzyme involved, the enzyme that carry out the carboxylation, this one is Rubisco, is the enzyme catalyze the carboxylation. Here, this is PEPKase, PEP carboxylase. You can write PEPKase also, is the uh, PEPKase case catalyze primary carboxylation reaction. Okay, this one is only C3 cycle or the Calvin cycle. Cycle is present in C3 plant. But in C4 plant, what happened? They have actual process for the carbon dioxide reduction is the Calvin cycle. So all the photosynthesizing plants, they have C3 cycle. So these C4 plants, they have C3 cycle as well as C4 cycle. So C4 plants have C3 cycle and C4 cycle, means 2, is it? Here another thing is the difference is there. In C3 plant, the light reaction and the dark reaction. Dark reaction particularly, that is the Calvin cycle, that takes place within the same type of cell, is it? The light reaction of photosynthesis and dark reaction takes place in the same cells, that is the mesophyll cells and chloroplast. In the same chloroplast, both light reaction, dark reaction. But here what happened, that light reaction in the mesophyll cell and light reaction takes place in mesophyll cell chloroplast, and dark reaction, that is Calvin cycle takes place in bundle sheath chloroplast. Okay. Next is number 6. They do not have C3 plant, no cranch anatomy in leaf. Okay. But the C4 plants have cranch anatomy present in leaf, leaf anatomy. Next important point is that this one, in the C3 plant, photorespiration occurs. Photorespiration takes place. But here in C4 plant, photorespiration is nearly nil. Is nearly nil. Those points also you can write that the high temperature is suitable, high temperature and bright light. Okay, optimum temperature is high. Here the in C3 plant it is low. It is lower light intensity. They can carry out photosynthesis. In C4 plant in high light intensity, that is nearly 35, 40 degree, they can carry out photosynthesis nicely. And then now come to the expenditure. Okay, for the synthesis of one molecule of simple sugar or simple carbohydrate, that is you can write glucose, fructose 18 ATP and 12 NADPH are required. But here, how many? For each molecule of carbohydrate to produce, they have they require 30 ATP and 12 NADPH, okay, are required. Optimum temperature is low. Optimum temperature is high. Optimum is that is at which temperature and at which level this any physiological process that occur maximum, okay, that is suitably the temperature which is suitable for any of the physiological process, that is the optimum temperature or optimum condition, okay. There one is called the carbon dioxide compensation point, okay. Here the carbon dioxide compensation point is very high. Carbon dioxide compensation point is high. Here the carbon dioxide compensation point is low. That is carbon dioxide compensation point here. Carbon dioxide compensation means it is say in the plant what happened? Photosynthesis taking place and also respiration. All right. This photosynthesis require carbon dioxide in and it produce what? Carbon dioxide is absorbed and oxygen is produced. This oxygen is utilized by this oxygen is utilized in the this oxygen is utilized in respiration, is it? And the respiration will produce the carbon dioxide, and this carbon dioxide is again utilized by the photosynthesis. So this things goes on. The product of the photosynthesis is the raw material for the respiration, and the product of that byproduct of the respiration, that is the raw material for photosynthesis. So carbon dioxide compensation point is that concentration of carbon dioxide. Compensation point is the concentration of carbon dioxide at which it can compensate the rate of respiration. That is, it can compensate that for the this photosynthesis will takes place, and that oxygen will be sufficient to carry out respiration, so that the plant will not absorb oxygen from outside. Okay, so this is very high in case of this. To reach this saturation, it is very high in case of C3 plant, but for the C4 plant it is very low. Means in low concentration also it can carry out the photosynthesis efficiently, and so it can compensate, it can produce enough oxygen which can support the respiration. Okay, so here it is, and there are many several other small points are there, but you should. This is enough. Many point I have given, so these things you have to remember. Okay, and what is so? Is there any question, dear students? So you will, this one is very, very important, this difference between C3 and C4 plant. Okay. Most of the plant, they are C3 plants. All right. C4 plants are very few. Okay. So, but the name I have given for the, this NEET exam, etc. Also, this one is very, very important. Like those, what are the, which one is the C4 plant, which one is C3 plants, then which has the high compensation or low compensation, how many this or how many ATP or NADP require. These are a small thing that based on that many questions are usually asked in the NEET exam. Okay, and for the board exam, of course, also it is very important. Okay, in the next class, today is this. Next class, I will talk about something about the photo respiration. Here, of course, today I have little bit um, explain you a little bit. I have mentioned, not explain totally. So in the next class I will do that photo respiration. Okay, you you solve the solve this practice paper and practice from the practice book, and you those for the board exam, you please give that which do you need any question to be solved. Note I'll give. I'm writing it, and I'll give you. Okay. So by today I will conclude here and stop. All right. Do you have any question, anything to ask?